So let's get some terms clear. There's kind of two types of topographic maps in the brain. There's retinotopy maps, which you all know, where nearby neurons or patches on the surface of visual cortex are sensitive to nearby small locations in the visual field, which maps onto the retina. And then there's another kind of topography related to functional specializations. So this is where nearby regions on the surface of the brain, the human brain in this case, prefer similar features having to do with visual properties other than locations. So in this case, sensitivity to faces versus buildings. So this is another example of topography. And what we're starting to gather evidence for, and is becoming more and more clear, is that these two types of topography, retinotopy and functional specializations, are related. So here on the right side, you can see the retinotopic map for the same region with foveal central regions in yellow and more parafoveal or peripheral regions in green. And the overlay shows you that the sort of face-sensitive regions are also more sensitive to the foveal visual field. So there's a relationship between these two maps. And this is what I mean by topographic variability. Different regions of the retinotopic map are sensitive to different visual features. And this is in contrast to how most people conceptualize and model coding in visual areas. The usual working assumption is that feature encoding is uniform across all locations of the visual field. And the advantage here, of course, is that then every location in the visual field has access to every feature. The disadvantage is that if your actual visual experience doesn't also have uniform feature statistics, then this is kind of wasteful. And to get a little more specific, most models of the visual system, the leading models, specifically CNNs, inherently don't and can't allow, in their versions now, non-uniform coding just because of weight sharing. So we assume that every pixel location has the same weight, has the same filter, has the same feature properties. So there's this disconnect between brains and models. And I should say I'm not the first person to make this observation. The OGs of functional specialization in the visual system, Mishkin and I always have to make sure I pronounce this, Ungerleider, very early on. So these are one of the first scientists that introduced the idea of the dual stream system. There's a what pathway and a where pathway, and these two physically separated pathways in the brain, they do interact with each other, but they have these biases for different functional features with different behavioral relevance. And very early on, this is an abstract from 1983, they noticed that these two pathways also had different biases for the visual field. So this is, again, that same topographic variability. And about a year ago, Patrick Minot, who's a lot of you probably know from Life, from his work, but also from Twitter, kind of lit a little bit of a match by saying that CNNs aren't all that much like the early visual system, in part because they can't allow for topographic variability. But this idea and its practice and implementation of uniform feature properties and measurements, models of the visual system, and also in the training data and benchmarks that we use to assess our progress, this remains pervasive. So I thought it's worthwhile to take a couple minutes and flesh these ideas and the data out again. So in the next few minutes, I'll just go over some flavor of the evidence in different animals for topographically variable or non-uniform feature encoding, and then I'll tell you about why it might be worthwhile for models of the visual system to include this biological observation. All right, so non-uniform encoding starts in the retina, and it's visible to the naked eye, well, under a microscope. You don't need fancy techniques to see it. In the retina, the density, the cell type, and the feature responses, the functional properties, of the ganglion cells that project to the rest of the visual system can vary pretty greatly across the surface of the retina. For primates and humans, the variation is greatest along this central peripheral axis. For rodents and some other small animals, there's not such a clear central peripheral distinction, but there is a dorsal ventral axis difference, so the upper and lower portions of the visual field seem to have different properties. And as you might imagine, some of these differences that originate in the retina are preserved or even get enhanced as we get to the cortex. So here in primate V1, the neurons in the central portion of the retinotopic map, I'm going to call these the central representation, prefer slower moving things, and the neurons in the peripheral representation prefer fast moving things. And this is in a study that specifically made sure, sort of did the hard job of going all the way out to the periphery, all the way out to 90 degrees and measuring these properties. Usually people subsample and only focus on the central regions. And it's not even just functional properties. Cell types in the cortex can also vary along eccentricity. So in the pathway from V1 to the motion area MT, there's a centricity, central periphery, dependent difference in the cell type in this connection pathway. And I think, and there's a couple of review papers, including our own, that talk about some other differences, including those in higher order visual areas like IT that I mentioned at the top. And I think all of this is still an undersampling of the topographic differences of non-uniform feature encoding across eccentricity in the primate, because we know from clinical studies where people have damage to either their central retina or their peripheral retina, that damage to these different portions has very different behavioral consequences. So presumably, if we tried to really get good sampling, we would find other differences. And we know more about non-uniform coding in the retina and visual system of the mouse because wide field measurements are easier there. So in mouse V1, there are a couple of differences along this elevation upper-lower visual field axis. In V1, color preferences changes across this axis, the sensitivity for coherent global motion, and also the sensitivity to binocular cues that the brain may use to assess depth in 3D. All of these vary along this elevation axis. Okay. And these are, these aren't minor differences. And this distinction between the upper and lower visual field can be seen within an individual area like V1, but I don't know if some of you might have noticed, also across the entire visual system of the mouse. So you may not know, but in the mouse visual system, using similar anatomical connectivity rules as the primate, people have used supervised and unsupervised techniques to define a dorsal stream and a ventral stream and assign the sort of ring of higher order visual areas that sit around V1 in the mouse to either of these streams. And although the feature specializations in the mouse dorsal and ventral streams aren't as huge as they are in the primate, there's no face region, there's no dedicated sort of motion area, they do exist where one of the streams sort of likes motion and maybe depth cues, and another one sort of likes texture and is more sensitive to color differences. But the really striking bit is that if you combine the little visual field from all of the areas in the dorsal and in the ventral stream, they divide really nicely along this upper-lower visual field division line. So this tells you that the visual system of the mouse has different functional and anatomical properties for the upper and lower visual field. And I mentioned in the intro, but the primate dorsal and ventral stream that famously have different feature sensitivities. It's kind of one of the first functional specializations that were noted in primate brains. They also have these visual field biases. So the dorsal stream receives input from nearly all portions of the visual field and uses this input for things like posture control, moving around, grasping, and reaching. The ventral stream receives biased visual input from mostly the foveal central regions. So single receptive fields in ventral areas like IT can be quite large, but overall there's a bias for the central visual field. And the relationship between the streams and their visual field bias, it's not as clear in the primate as it is in the mouse, but there is a visual field bias in both animals, and it's different. In primates, it's the central peripheral axis. In mice, it's along this lower and upper visual field axis. And so why might this be the case? Why would these axes differ? Why would there be a bias in the first place? A nice, easy story to tell is that this non-uniform coding is an efficient representation of non-uniform statistical regularities in the natural visual experience of animals like social primates who maybe face each other routinely. So the central bits on the retina look different than the peripheral bits. But then for mice that sort of sit lower on the ground and have to really watch for predators in the sky, there's salient statistical differences between the upper and lower visual field axis. But this is just, this is another, just so evolution story until it's tested. So that's the data. Why bother incorporating it into models? After all, abstractions and simplifications are necessary. The trivial answer is to improve biological plausibility and get more brain-like representations, but that's not all that relevant to me. What is relevant is having a model that can go beyond object recognition. So our models are good right now, but that's in part because our benchmarks and our training data sets are all sort of somewhat focused on object recognition in the central visual field. If we want to add these other abilities, hunting, eating, navigation, peripheral vision and its distinct feature sensitivities are going to have to come into play. And again, we know this from clinical studies. We could also gain species-specific organization that would come in handy for modeling, for instance, the mouse visual system, because it's increasingly where we make our measurements that can be used to verify and improve the models. It's hard to imagine that ImageNet and these other training data sets used for human object recognition would serve well for the mouse. But we can get a bit closer to imitating the statistical regularities of the mouse's visual system by imposing known non-uniformities measured in V1, and it turns out the retina in mice also has the same color preference. It's actually, the V1 preference is actually inherited from retina. We can impose these as priors and get a little bit closer to having useful visual inputs for these other species models. But it's even better to start with the visual experience and extract the non-uniformities by looking for the statistical regularities that can then serve as ecologically relevant priors for the visual system. So Tom Euler's group did this. I'm not going to play the video, it doesn't matter. It's basically, this is a mouse's view navigating its environment, and they measured the intensity of UV across the visual field, ultraviolet, green, and this is the combination of both. And when they plotted out those intensities across a function of elevation, they saw distinctions that matched what we see in the retina and what we see in V1, and also what we see in the behavioral reports of the animals. All right, so we could do the same thing, and we could also analyze these animal point of view videos for lots of other statistics. And coupling non-uniform feature encoding with animal point of view data sets would really sort of open the door and allow us to determine what other aspects of natural embodied visual experience explains the various non-uniformities and organizing principles in the visual system of primates and rodents. So for instance, right now, if I asked you, how should the visual system of a primate differ from that of a rodent? If I tell you the differences in their ecologies and their environments and the differences in how they move and in their eye positions, most of us wouldn't get very far. But if we add non-uniformities into our models and improve our training sets so they really capture the visual experience of animals, we might be able to answer these questions. So I'm over time. There's a couple of ways to include these non-uniformities, and Shahab, my co-author, and I, he's at Mela McGill with Blake Richards, are starting